Okay, so my name is Stefan Przyborski. I am the Chief Scientific Officer of Reinnovate. I'm also a professor in cell biology at Durham University, and I developed Alvetex technology as a solution to three-dimensional cell culture. Today, we're going to be talking about 3D culture and its focus when using and the development of co-culture models. So during this presentation, I'll first cover the limitations of 2D culture, and I'll follow this by an introduction to 3D and our Alvetex technology. We'll then look at a series of applications of where Alvetex has been used and include instances where co-culture has been developed. We'll then finish off by describing methods that are compatible with Alvetex technology. Okay, so in the normal situation in the body, in vivo, cells grow in tissues as shown in this histological section. And we can show these as well by this schematic diagram. And we see an individual cell with its extracellular matrix against adjacent cells. In vitro, we break up these relationships and cells enter a synthetic environment such as a petri dish as shown here, or the multi-well plate, etc. And when cells come into contact with this foreign synthetic substrate, either glass or polystyrene plastic, cells adapt and they flatten out and form abnormal morphologies. Now, as the cell remodels, its cytoskeleton adapts to these environmental changes, and this in turn triggers changes in response to gene expression and protein translation, and collectively cells behave differently in terms of their function in two-dimensional environments as compared to their three-dimensional counterparts. Let's consider this a little more closely in 2D. So here we see that almost half the cell is against the plastic substrate in this petri dish. Oops. The other half of the cell is mostly against the culture medium. Cells are flattened. Interactions between cells are minimized, and certainly in 2D culture there's very limited capability of creating more organized structures as what we see in vivo. If we look at a single cell by confocal microscopy, we can see very clearly how in 2D a cell becomes exceptionally flattened, whereas in 3D its structure is 3D all the way around. This is a fibroblast which has been stained with floydin, illuminating the F-actin cytoskeletal component using confocal microscopy. Similarly, organelles in the cell, such as the nucleus, are also changed in structure. And here we see how in 2D the nucleus is very flattened, and in 3D it maintains its spherical shape and form. And in the primary literature you'll see many examples of where alteration of the cell shape influences gene transcription, protein translation. So this is a phenomenon which has been known for a long time. So we're trying to prevent that shape change from occurring when growing cells in the laboratory. And we've created a solution for simple and routine three-dimensional cell culture that recreates the natural three-dimensional environment for cell growth. So what we see in these two images here and here are histological sections of cells growing either in 3D or in vivo when transplanted into an animal. And these are exactly the same cell type. This is grown in Alvetex, and here grown as a xenograft. And that's what this technology is all about. It enables you to grow cells now in 3 dimensions in a way that more closely mimics the structure of real tissues in the body. So we have evidence of genuine 3D cell culture, increased cell viability and enhanced functionality, and as I say, this technology can create situations which are more representative of real tissues. So let's introduce this technology. It's called Alvetex Scaffold, and it is a polystyrene scaffold. So across the top line here, we have our 2D situation where we have our vessels, which you'll be familiar with, such as the Petri dish, 2 dimensions, cells go to the model layers, as we can see here. In this situation with Alvetex, we have re-engineered polystyrene to create a highly porous polystyrene scaffold into which cells can enter and grow in 3-dimensional structures such as these. The scaffold is engineered into a 200 micron thick membrane. You can see that in transverse section here and at high magnification here. And cells enter the scaffold, and we can see that this scaffold here is full of cells, and you've created a in vivo-like structure, almost like a slab of tissue inside the material. These are the various formats of Alvetex which are currently available. These include multi-well plates, such as the 12, 24, and 96 well versions. And here we have well inserts. We have available currently 6 and 12 well inserts. The presentation of the scaffold is really very important for how the cells grow and function inside the material. So in scenario A, we have a situation where the scaffold is presented on the base of a multi-well plate, such as the 12 or 24 well plates, and medium supplementation from above only. In the well inserts, we have medium supplementation from below and above, as you can see. In scenario C, we've developed this product, and this is a cradle which sits inside a Petri dish and houses 3 well inserts. And we've developed this so we can have a larger volume of medium to supplement the growth of the 3-dimensional cultures whilst they're growing inside the Petri dish. And that's useful for longer term culture. Now, Alvetex is a very versatile technology, and it can be used for multiple applications in different ways. And one of its strengths is growing cells in coculture in 3 dimensions. This slide shows a variety of different scenarios whereby cells can be grown in conjunction with other cell types within our product. So here we've got a disc of Alvetex scaffold on the base of a multi-well plate, and we've seeded into that 2 cell types simultaneously. So that can be done inside a plate or inside a well insert. In this scenario, we've got a 3-dimensional culture of a single cell type in the well insert and a 2-dimensional culture of a second cell type on the base of the well on a standard plate. And here we've got therefore signaling capability between the 2 cells via paracrine signals. This can also be done in 2 independent 3-dimensional cultures as shown in this scenario. In this situation, we have 2 separate 3D cultures, but actually they're in direct contact with one another. And this is a useful situation for studying how cells in different layers could have impact on each other via either migration or direct paracrine signaling. And in the last scenario, we have a 3D culture on the base and then a 2D culture of cells seeded on the surface. And I'll show you examples of some of these as we go through this presentation. So Alvetex has been used for many different applications, and this is work which has been completed by ourselves, our in-house scientists, but also by our customers and through multiple collaborations. And there is a wealth of data to demonstrate that Alvetex influences the structure and function of cells, their growth and differentiation, how we can use this technology to develop assays and novel in vitro models, and how we can create organized tissues and develop sophisticated organotypic systems. So let's go through a series of examples. Number 1 is focused on liver. So first of all, I'll talk about the growth of hepatocytes inside Alvetex alone, and then finish with a coculture situation. In this slide, we see primary hepatocytes here, which have been labeled with a fluorescent dye and then imaged by confocal microscopy. And we see how the cells are flattened in 2D culture compared to those in 3D, which retain their native cuboidal shape and form. And these cells have been seeded purposefully at low seeding densities to enable individual cells to be imaged. Here we have primary cells growing in the top part of the Alvetex scaffold. These are primary hepatocytes, whereas here we have HepG2, a well-known hepatocyte cell line, and we're showing histological images here, and scanning electron microscopy images of those cells here. This technology is also amenable to transmission electron microscopy and immunocytochemistry. So in this example, we are seeing two cells coming together, tight junctions here and here, and between we've got a biocannuliculus forming between this hepatocyte and that hepatocyte. And we can see microvilli protruding into the lumen. And we can identify these structures further by staining for proteins expressed on the apical membranes of these cells, such as this molecule here, P-glycoprotein. And we can see that illuminated there very nicely, that pink staining. And certainly many of our data are published in peer-reviewed scientific publications. With primary hepatocytes, we notice an enhancement in cell viability. So if we look at 2D control and 3D Alvetex scaffold, and we determine the number of live, dying, and dead cells at different seeding densities, we can clearly see we have a greater proportion of live cells compared to the dead and dying, much more so than in the 2D situation. In addition, the metabolic function of hepatocytes is also enhanced in Alvetex scaffold. So in this experiment, hepatocytes were grown and then exposed to a cocktail of model toxins for up to 48 to 72 hours, and then the expression or the induction of these enzymes, these P450 enzymes, 1A2, 2B1, and 3A2, were determined and measured. And you can clearly see, looking at the levels of either the 3D in black or the 2D in grey cultures, that there's a significant increase in the functionality and induction of expression of these enzymes in 3D culture compared to 2D culture. A similar assay was done in this experiment, where we're looking at the expression of CYP3A4 activity. And this work was done in collaboration with Medicite. Medicite have a population of human upside hepatocytes, which are genetically modified, and this enables them to proliferate. These are primary cells, and they can proliferate for up to 40 population doublings. And these are used to assess drug toxicity and study metabolic function of liver cells. In these experiments, these cells were grown either 2D plates or 3D Alvetex plates, either in isolation or in co-culture with endothelial cells. The data clearly show that there's an induction in CYP3A4 activity when growing in 3D, but also a further induction with the presence of the endothelial cells. And these experiments were performed by mixing the cells directly together and co-seeding them into the Alvetex scaffold. So here's a second example, and this refers to a model we've developed for studying neurite outgrowth and neural differentiation. And we're using this model to investigate the mechanisms that are responsible for neurite inhibition during the formation of the glial scar in spinal cord injury. So we generate neurons, human neurons, from human pluripotent stem cells, and we create neurospheres. And this is an example here of a neurosphere which has been plated upon a substrate coated in laminin, so it's a 2D substrate, and these neurites will grow out and radiate, such as this. This neurosphere structure is quite substantial. It's about 500 microns or so in diameter. So these are quite substantial neurites. We can take these structures and we can also place them on top of Alvetex scaffold. So this rectangle here represents an Alvetex disc, and you can visualize cells from either the top, the side, or the bottom using fluorescence microscopy. So there's the top elevation, looking down on top of a neurosphere. We can see the neural pericardia, and here we have green TUJ1 staining for the neurites. From the side, you can see up here at the edge of the scaffold, these are primarily where the neuropericardia are located, and these are neurites going into the scaffold. And the bottom elevation, so this is looking underneath here, these are neurites which have journeyed all the way through the 200 microns of porous polystyrene scaffold. And we can count how many of these are present and measure their lengths. So in the experiment, we can begin to study human stem cell derived neuritogenesis and understand the mechanisms by which those neurites respond to molecules expressed by reactive astrocytes. So during a spinal cord injury, a compression of the cord can result in activation of the astrocytes, which become reactive, and they express proteoglycans around them. Aggrecan is a protein of such an example, and we can take aggrecan and we can coat Alvetex with this protein, and this is what this symbolizes here. We can then grow our cells as before, and we can study how those neurites grow in 3D in response to aggrecan, and if there's any inhibition, we can then look to see if we can recover that inhibition by adding various small molecules. So let's look at some data. So here we have a side elevation, once again, of control. So here are the neurites, here are the cell pericardia, the cell bodies. Notice you get ingrowth, but in a scaffold coated with aggrecan, the amount of neurite outgrowth into the material is suppressed. And you can see this very clearly on the bottom elevation, so control, neurites, aggrecan coated, very few neurites. But if we add a small molecule such as this retinoid agonist, so this is an RAR beta agonist, so this is a retinoid, this molecule can overcome that inhibition effect resulting from aggrecan coating. And we can quantify these data very nicely by looking at the number of neurites in the different conditions, and we can show statistically that there is indeed a difference between the controls and the fact that aggrecan control can be rescued when we add the retinoid, and also we can rescue it using this other small molecule, which is Y27632, and well-known ROCK inhibitor. So we can now take this to the next level, where we can do co-culture, and we can actually study the cells that express these types of protein, these negative regulators of neurite outgrowth, to try and mimic the glial scar in vitro. So in these experiments, we've co-cultured human stem cell derived neurons with glial cells, and we can see that at low magnification in panel A. In panel B, higher magnification. So we've got the glial cells in here, and here are the cells in the neurosphere. So notice in C, okay, this is the control, so this is a monoculture, it's only the neurosphere. We've got nice neuro outgrowth viewed from above, just as before. And in D, all of these DAPI stained nuclei are of the glia, but notice now how the neurites tend not to grow out, one or two do, but the majority tend to wrap around the mass of neuronal cell bodies. We can also look at this from beneath, just as before. So in the control, urites come through all the way. So this is in monoculture. There's no glial cells in panel A. In co-culture, okay, so these are the DAPI nuclei here of the glial cells. And if we add, again, this ROCK inhibitor, Y276C2, we get rescue, okay, of neurite outgrowth. So none in co-culture, and then rescue in the co-culture situation when we add the ROCK inhibitor. And again, we can quantify that as shown beneath. So this is a nice model of how one can co-culture two different cell types in 3D and then perform a quantitative assay. In this way, it's neurite outgrowth. Okay, so here's a different example. This is modeling cancer cell migration and invasion. So what I'm showing here is a pathological sample of a colorectal tumor. This is the tumor. This is the wall of the colon. You can see the crypts here, and this is muscularis. So these cells are beginning now to invade the surrounding stromal tissues. To study this in vitro, we have two well-known cell lines, SW480 and SW620. And these represent pre- and post-metastatic stages of the disease, respectively, and they are isolated from the same patient. The 620 cells have a lower E-cadherin expression and higher vimentin, and therefore one would predict that they are more predisposed to migrate and invade more than the 480 cells. And indeed, if we feed the cells onto the materials, we will see how, on average, the SW620 cells penetrate to a greater extent compared to the 480. And that can also be recorded on this plot and quantified as so. So I mentioned the fact that the tumor cells are invading the surrounding tissues, and we can now consider how we can use our Alvetex technology to study this process. And we've been doing this for a proof of concept study involving co-culture of fibroblasts with the tumor cells. And as a proof of concept, as I say, initially we've been using 3T3 fibroblasts to mimic the stromal tissue. So first of all, we create, over 7 days, a three-dimensional culture of fibroblasts, such as this, and then we seed the colon cancer cells on top to create co-culture models such as these. So in this example, we've got 480 cells here and 620 cells here, and these are the fibroblasts involved. So as I say, these are 3T3s initially, but we've now got access to cancer-associated fibroblasts, so we can build a much more relevant model. To distinguish between the cell types, one can perform various assays. One can have one cell type which is carrying a fluorescent reporter, such as GFP, and we now also have access to some GFP-labeled fibroblasts, and we're now working on those. But also it may be that the two different cell types express distinct markers or proteins, which one can then subsequently detect by immunocytochemistry. In conjunction with studying cells and looking at their invasion, we can also manipulate that migration of cells through the stromal layer, and we are working on looking at doing this by adding various chemoattractants. So in this scenario, we've got the well insert sitting in a well of media, and we're adding to the bottom chamber a chemoattractant. And we've been using IGF-1, and you can see here, in this example, 1 nanogram per milliliter, and we're looking at the penetration, okay, migration in this case, of these two cell types into the material, and that is being measured, the average distance measured into the material has been determined and then quantified, and we can see a significant difference in the migration of SW480 cells in response to IGF, okay, but not SW620 cells. Now that's all histological data. Now if you don't want to do histology, it's also possible to measure cell penetration into the material using confocal, and this is work which is very kindly shared by Patricia Muller from the Beatson Institute in Glasgow, and she is showing here in the control situation and in the experimental situation. This is a cell line carrying a P53 mutation, and these are optical sections through the material up to 90 microns into the material, and you can see clearly how those cells carrying the mutation migrate more deeply into the Alvetex compared to the control situation. Okay, so another example is a skin model that we've developed. So this is the culture of skin keratinocytes and dermal fibroblasts in Alvetex. And the objective is to produce a differentiated skin with a stratified epidermis and cornification at the surface to create what is known as the stratum corneum. And this is what the arrows are pointing towards here. So these are two different examples. We have Alvetex, first of all, impregnated with dermal fibroblasts. This is a primary cell type, human in both examples. That is established first prior to the seeding of keratinocytes on the surface, and then bringing the culture up to the air-liquid interface to result in the stratification of the top layers, i.e. the formation of the stratum corneum. We can follow that process nicely using immunotouch chemistry. So here's a schematic showing the various proteins expressed as these keratinocytes migrate from an immature state to the surface and mature and differentiate, forming the cornified layer. And you can see that various markers are expressed. Keratin 14 expressed in the bottom. You can see that in the bottom here of this culture, whereas 10 is more mature, higher up, and involucrin is a mature marker of the top layers. You can see that on the very surface. We could also study the differentiation of these cells using transmission electron microscopy. So we've got very nice examples here of the cells beginning to lose their nuclei as they go towards the surface. The number of desmosomes, these little black dots here, are the tight junctions between cells, and they begin to fall away and decrease as the cells move towards the surface, as the cells prepare to be lost from the surface. And we can see those specializations between cells, these tight junctions, very nicely in the higher magnification images at the bottom. We're particularly interested in those top cells, the cornified envelopes, and we've been studying the differentiation of those cells using a cell line. This is known as a Hackett cell line, and we can prepare these cornified envelopes from our cultures using an aggressive methodology involving boiling samples in SDS. And what remains are the ghosts of those cells, which are full of lipids and ceramides, and they can be stained with lipophilic fluorescent dyes. So we can see here, if you do that in a 2D culture, you get very few of these cornified envelopes formed, whereas in 3D you see a far greater number, and these are the cornified envelopes from real skin, which are comparable to those we are viewing in 3D culture. And the last model I want to show you is the development of epithelial and mucosal systems using our technology. So many epithelia are, by their very nature, monolayers of cells. You think of the basic simple epithelia in particular. So ideally, they would not grow very well inside the scaffold. So what we can do is apply a procedure to coat the surface of the scaffold with a very thin gel. This is actually a gel of collagen 4, and you can see that in this schematic diagram, and you can see the collagen gel stained there with this protein dye. See as well inside the insert how homogeneous the distribution this dye is and the coating of the gel. So it's all the way across the material. We can then, on the surface of the collagen, seed the epithelial cells of interest. And these are CACO2 cells. This is a cell line from the colon, and you can see a very nice monolayer of cells all the way across the material. So you can see the cells on the top, you can see that gel, and you can see the empty scaffold beneath. Now, you may say, well, I can do that in a transwell. That is true, but transwells are not especially porous. They are at best 30% porous. Alvetex is 90% porous, so you get much better of exchange across that membrane than you do a transwell. In addition, growth of these epithelial cells on extracellular matrix proteins such as these is important and much more physiologically relevant than a PET membrane. So let's look at those cells in a little more detail. So here are the cells again. This is actually a resin section stained with toluidine blue. And now we're looking here at floydin. So these are the microvilli. You can just see the microvilli there, and you can definitely see them here under TEM. And what we have here is staining for occludin, which reveals the apical tight junctions. So these structures here. So we can see those really very nicely, and we can see those little dots around the surface. And there is an example as well as of a microvillus at higher magnification. So that's the epithelium I just described, sitting on the surface of an equivalent to the basement membrane, this layer here, containing extracellular matrix proteins. Beneath is the laminar propria, which is a complex tissue consisting of various cell types, capillaries, fibroblasts, lymphocytes, etc., and the ECM. And we can model that inside Alvetex to create, with the epithelium, a mucosa, a full-thickness mucosa, such as this. This is obviously a schematic, but we've done this with fibroblasts and CACO2 cells once again. So here are the CACO2 cells on the surface, resting on top of, in this case, a collagen 1 layer, and here we have fibroblasts. And these are fibroblasts derived from the large gut, okay, the colon. So it's a true coculture model where we have a relevant stromal cell type beneath and a relevant cell line above. And this can then be used in various ways, for example, studying the relationships between these two cell types in health and disease, but also the functionality of that epithelium in terms of the transportation of solutes and ions across that epithelium, perhaps using a nushing chamber model. So if you're developing these models in well inserts, it's particularly useful because the well insert is a two-part assembly. It snaps open, the disc and the 3D culture such as this can be then mounted into an ushing chamber, and then short circuit currents can be measured. So the evidence I've shown you shows that the growth of cells in Alvetex in 3D culture influences their structure, function, viability, differentiation, and organization. Interestingly, Alvetex can be used in other ways. So here's an example of taking primary tissue and putting it on the surface of the scaffold, and the cells flow into the scaffold. We can see them now maintained in 3D, so they never encounter a two-dimensional environment. You can also use the material to support the growth of tissue materials and other structures such as this embryo body. So this is an embryo body derived from human pluripotent stem cells maintained on Alvetex. And we're developing this technology and application is an alternative for teratoma assay, where we can take these structures, we can then stain them for the markers of the 3 germ layers, and we can detect all 3, and we can then perhaps use that as an alternative to growing cells in an animal. So let's finish off by just covering a few technologies and analytical methods that are compatible with Alvetex technology. So one of the questions we often get is, how do we visualize cells inside the material using a conventional microscope? So we've created a very simple staining process. We use this dye neutral red, whereby we add that to our culture, and it's non-cytotoxic. The cells turn red, you can see the distribution very clearly, and under the microscope you can see individual cells and get an idea of distribution, viability, and density of the culture. And this can be used initially during the optimization stages of getting used to using Alvetex technology. Thereafter, it is not necessary. So growth of cells in Alvetex is just like growing tissue, as I've already described, and like tissue, one can fix the cells. So these are discs of Alvetex fixed with buoyants, and it's a yellow color. You can see how over 4 to 21 days the intensity of yellow increases. Those discs are then embedded in paraffin wax, just like tissue, and then sectioned to create sections mounted on microscope slides, and these are then stained with, in this case, hematoxylin and eosin. So you can see the culture beginning to form over 21 days. And under the microscope you can see 3D cultures such as this. So our strength in terms of 3D cell culture technology is the formation of layered structures as they would form in the body. Once you create those sorts of slides, you can then do immunocytochemistry, whereby you can visualize the expression of proteins, such as, in this case, Ki-67. This is also possible in frozen sections. Some antibodies don't like fixation and paraffin wax, therefore this can be performed using OCT. Confocal, as I've already described, and light cell imaging is possible, as is electron microscopy, both transmission and scanning electron microscopy. Just like standard cell culture plastic, Alvetex can be coated with various reagents. You'll see some examples listed here. These are all very familiar. And we see here a nice example of collagen 4 creating a 3-dimensional web of collagen filaments in the material. I've already mentioned how we can coat the surface, create thin layers, and put other cell types on top. It's also feasible to transfect the cells in Alvetex directly. We work in partnership with a company called Neurospheres based in the US, and they've developed bespoke transfection reagents, and here we have examples of GFP expression, and over here luciferase. Alvetex is also compatible with a vast majority of commercially available kits. So we have a kit here for MTT to measure cell viability, also MTS, XTT, Alamar Blue, Cell Type to Glow are all compatible with Alvetex. It's also feasible to isolate protein and nucleic acid directly from the 3D culture without having to isolate the cells, and the lysis buffers from these commercial kits can be added to the 3-dimensional cultures and the supernatants collected. And lastly, it's possible to determine cell number in Alvetex by using a picogreen assay that measures double-stranded DNA. And this works by determining cell number against fluorescence activity in a standard 2D culture. Creating that standard curve can then be used to read off values from absorbance from 3D cultures to determine the number of cells in the 3D system. Lastly, I just want to draw your attention to the fact that our website has significant online technical support, where you can learn about setting up your cultures and how to monitor and analyze them subsequently. Included in this are some instructional videos, such as those shown on this slide. So the final slide is a summary whereby we recognize that Alvetex provides a versatile platform for routine three-dimensional cell culture. It is easy to use, it is widely available, and its use is becoming more widespread. Scientists are now presenting their findings using this technology at scientific meetings and publishing their results in peer-reviewed journals. For further information, please do visit our website at renovate.com. In addition, if you have any questions, please do contact our technical support team at techsupport@reinnovate.com, and many thanks for your attention. Thank you.